Agricultural justice, gene editing and the fight for food sovereignty. This article provides a summary of its contents in the following manner. 1. Introduction. 2. What is gene editing? 3. Benefits of gene-edited food. 4. Concerns about gene-edited food. 5. Current status of gene-edited food regulation. 6. Conclusion. 1. Introduction. Gene-edited food is a topic that has captured the attention of scientists, policymakers, and the public alike. With the potential to increase crop yields, reduce food waste, and decrease the need for harmful pesticides, gene-edited food has been hailed as a promising solution to global food insecurity. However, it is also a controversial topic, with concerns about the environmental and health risks associated with genetically modified organisms. Whether you're a fan or a skeptic, one thing is clear: gene-edited food is a topic that demands attention and discussion. Gene-edited food refers to crops or animals that have been genetically modified using a technique called gene editing. This involves making precise changes to the DNA of an organism using various techniques, such as CRISPR-Cas9, in order to create desired traits such as resistance to pests and diseases, longer shelf life, and increased yield. Unlike traditional genetic engineering, gene editing can make targeted changes to existing genes rather than inserting foreign genes. However, gene-edited food is a controversial topic, with concerns about potential environmental and health risks, and ongoing debates around safety and regulation. The controversy surrounding gene-edited food is multifaceted and complex. On one hand, proponents of gene-edited food argue that it has the potential to address global food insecurity by increasing crop yields and reducing waste, while also reducing the need for harmful pesticides and herbicides. They believe that gene editing is a more precise and predictable way of modifying an organism's DNA than traditional genetic engineering, and that it can be used to create crops and animals that are more resistant to pests and diseases, have longer shelf lives, and require fewer resources to produce. On the other hand, critics of gene-edited food are concerned about the potential environmental and health risks associated with genetically modified organisms. They worry that gene editing could have unintended consequences, such as creating new allergens or disrupting ecosystems. There are also concerns that gene-edited food could have unintended consequences for human health, such as the potential for gene editing to create antibiotic-resistant bacteria. Another aspect of the controversy surrounding gene-edited food is related to regulation. Some critics argue that gene-edited food should be subject to the same strict regulation as genetically modified organisms, while others believe that gene-edited food should be exempt from regulation altogether. The controversy surrounding gene-edited food highlights the complex and often contentious relationship between science, technology, and food production. While gene editing has the potential to address some of the world's most pressing food-related challenges, there are also legitimate concerns about its potential risks and unintended consequences. 2. What is gene editing? Gene editing is a powerful and rapidly evolving technology that allows scientists to make precise and targeted changes to an organism's DNA. One of the most widely used gene editing techniques is CRISPR-Cas9, which stands for Clustered Regularly Interspaced Short Palindromic Repeats in CRISPR-associated protein 9. CRISPR-Cas9 works by using a molecule called RNA to guide a protein called Cas9 to a specific location in the DNA of an organism. Once Cas9 has reached its target, it can then cut the DNA at that location. Scientists can then use this process to either remove or replace specific genes or gene sequences. The CRISPR-Cas9 system has revolutionized gene editing by making the process faster, more precise, and more affordable. It has been used in a wide range of applications, including in agriculture to create crops that are more resistant to pests and diseases, and in medicine to develop new treatments for genetic disorders. While CRISPR-Cas9 is a powerful tool, it is not without limitations. One challenge is the potential for off-target effects, where the Cas9 protein accidentally cuts the DNA in the wrong location, which can lead to unintended consequences. Scientists are actively working to develop new and improved gene editing techniques that can address these challenges and further advance the field of gene editing. Gene editing and traditional genetic engineering are both methods of modifying an organism's DNA to achieve desired traits. However, there are some key differences between the two techniques. Traditional genetic engineering involves inserting foreign DNA into an organism's genome to create a new trait. For example, scientists can insert a gene from one organism into the genome of another to create a plant that is resistant to herbicides. This process can be imprecise, as it may not be possible to predict exactly where the foreign gene will be inserted, and it may have unintended consequences. In contrast, gene editing is a more precise way of modifying an organism's DNA. Rather than inserting new DNA, gene editing allows scientists to make targeted changes to existing genes. This can be done by cutting the DNA at a specific location using enzymes like Cas9, and then allowing the cell's natural DNA repair mechanisms to make the desired changes. This technique is highly precise and can be used to create more subtle changes to an organism's genome, such as turning off specific genes. Another important difference between gene editing and traditional genetic engineering is the level of regulatory oversight that each technique receives. Traditional genetic engineering is generally subject to strict regulation, and new genetically modified organisms must go through a rigorous testing and approval process before they can be released into the environment. Gene editing is currently subject to less regulation, although this may change as the technology becomes more widely used. Overall, gene editing offers a more precise and controlled way of modifying an organism's DNA compared to traditional genetic engineering. While both techniques have their advantages and limitations, gene editing is rapidly becoming an important tool for improving food security and addressing other global challenges. 3. Benefits of gene-edited food. One of the major benefits of gene-edited food is that it has the potential to increase crop yields and improve food security, particularly in regions where crops are vulnerable to pests, diseases, and extreme weather events. By editing the genes of crops to make them more resilient to these challenges, farmers can increase their crop yields, which can help to meet the growing demand for food in a rapidly expanding global population. This could have significant implications for developing countries, where food insecurity is a major challenge. Another potential benefit of gene-edited food is the ability to improve the nutritional content of crops. For example, scientists have used gene editing techniques to create rice that is fortified with additional vitamins and minerals, which could help to address micronutrient deficiencies in populations that rely heavily on rice as a staple food. Gene editing can also be used to create crops that are more resistant to herbicides and pesticides. By reducing the need for these chemicals, gene-edited crops could help to reduce the environmental impact of agriculture, including the pollution of waterways and the loss of biodiversity. This could have significant implications for the long-term sustainability of global food systems. In addition to these benefits, gene-edited food can also be designed to have better taste and texture, making them more appealing to consumers. For example, gene editing can be used to create fruits that are sweeter or vegetables that have a better texture, which could help to increase their consumption and reduce food waste. Despite these potential benefits, there are also concerns about the safety and ethical implications of gene-edited food. Critics argue that not enough is known about the long-term effects of gene editing on human health and the environment, and that more research is needed before these crops are widely adopted. Gene-edited food has the potential to address many of the challenges facing global food systems, including food security, malnutrition, and environmental sustainability. While it is important to continue to study the potential risks and benefits of gene editing, this technology offers a promising tool for creating a more sustainable and equitable global food system. 4. Concerns about gene-edited food. Gene editing technologies like CRISPR-Cas9 can produce unintended genetic changes in organisms, and there is a risk that these changes could have negative environmental impacts. One potential environmental risk of gene-edited foods is that they could become invasive species. If a gene-edited crop or animal is introduced into a new environment, it may have a competitive advantage over native species, and could potentially outcompete or even replace them. This could lead to a loss of biodiversity and disruption of local ecosystems. Another risk is that gene editing could lead to the creation of superweeds or superpests. If a gene-edited crop is designed to be resistant to a particular herbicide or pesticide, there is a risk that the weeds or pests it is designed to target will evolve resistance to that chemical over time. This could create more resilient and difficult to control pests or weeds, which could have negative implications for farmers and the environment. There is also concern that gene-edited foods could have unintended effects on non-target species. For example, if a gene-edited crop produces a new toxin, it could harm beneficial insects or other organisms that are important for ecosystem health. Finally, there is a risk that gene editing could lead to unintended effects on soil health or water quality. For example, if a gene-edited crop produces a toxin that is leached into the soil, it could harm soil microorganisms or other organisms that live in the soil. Similarly, if a gene-edited crop requires more water than traditional crops, it could have negative implications for water resources. It is important to note that these potential environmental risks are speculative at this point, and more research is needed to understand the long-term impacts of gene-edited foods on the environment. However, it is important that these risks are considered and addressed as gene editing technologies are developed and deployed. One potential health concern associated with gene-edited foods is the possibility of creating new allergens. Gene editing can involve introducing new genes into a food source, or altering existing genes, which could potentially lead to the production of new proteins that trigger allergic reactions in some individuals. For example, if a gene-edited crop is altered to produce a protein that is similar to one found in a common allergen, such as peanuts or shellfish, it could potentially cause an allergic reaction in individuals who are allergic to those foods. Additionally, if a gene-edited crop is modified to produce a new protein that has not been previously encountered in the food supply, it could be difficult to determine whether that protein is safe for human consumption. Another potential health concern is the potential for unintended effects on human health. While gene editing technologies like CRISPR-Cas9 are highly precise, there is still a risk that unintended genetic changes could occur during the editing process. These unintended changes could have negative effects on human health, such as increasing the risk of cancer or other diseases. It is important to note that the risks associated with gene-edited foods are still being studied, and more research is needed to fully understand the potential health impacts. However, it is clear that careful consideration and regulation of gene-edited foods will be necessary to ensure their safety and protect public health. Gene editing technologies like CRISPR-Cas9 have significant ethical and social implications, as they raise questions about the boundaries of human intervention in the natural world and the potential consequences of manipulating the genetic code of living organisms. One of the key ethical concerns surrounding gene editing is the potential for unintended consequences and long-term effects on human health and the environment. Gene editing can involve making changes to the genetic code of organisms, and there is a risk that these changes could have negative impacts that are not fully understood or anticipated. For example, modifying the genes of humans or animals could have unintended consequences for future generations, and could potentially create new diseases or conditions that are difficult to treat. Another ethical concern is the potential for gene editing to exacerbate existing social inequalities. If gene editing is only available to the wealthy or privileged, it could further widen the gap between rich and poor and create a society where only a select few have access to the benefits of genetic manipulation. Additionally, there is concern that gene editing could be used to perpetuate discrimination based on race, ethnicity, or other factors, leading to a society where certain groups of people are considered superior or inferior based on their genetic makeup. There are also ethical concerns surrounding the use of gene editing in agriculture and food production. For example, gene editing could be used to create crops and animals that are more resistant to disease or produce more food, but it could also lead to the concentration of power and resources in the hands of a few large corporations. Additionally, there is concern that gene editing could be used to create novel organisms that are not found in nature, raising questions about whether these organisms have the same rights and protections as other living creatures. The ethical and social implications of gene editing are complex and far-reaching, and will require careful consideration and regulation as these technologies continue to develop and become more widespread. 5. Current status of gene-edited food regulation. International regulations on gene-edited foods vary widely, and there is currently no universal agreement on how these technologies should be regulated. In some countries, such as the United States, gene-edited foods are regulated on a case-by-case basis, depending on the specific characteristics of the product and the methods used to create it. The U.S. Food and Drug Administration, FDA, has stated that gene-edited foods are subject to the same regulations as conventionally bred crops, as long as they do not contain foreign DNA and do not pose a threat to human health or the environment. In Europe, however, the regulations surrounding gene-edited foods are more restrictive. In 2018, the European Court of Justice ruled that gene-edited organisms should be subject to the same regulations as genetically modified organisms, GMOs, which are tightly regulated and subject to rigorous safety assessments. This decision has been controversial, as many scientists and industry groups argue that gene-edited foods should be treated differently than GMOs, as they do not necessarily involve the introduction of foreign DNA into an organism. In other countries, such as China and Brazil, regulations on gene-edited foods are still evolving, and it is not yet clear how these technologies will be regulated or whether they will be subject to the same regulations as GMOs. The regulation of gene-edited foods is a complex and rapidly evolving issue, and it will be important for international organizations and regulatory bodies to work together to establish clear and consistent guidelines that ensure the safety of these products while also promoting innovation and progress in the field of gene editing. There are several regulatory challenges associated with gene-edited foods, including the lack of a consistent international framework for regulating these products, the difficulty of distinguishing between gene-edited foods and GMOs, and the need for robust safety assessments to ensure that these products do not pose a threat to human health or the environment. One of the biggest challenges facing regulators is the lack of a consistent international framework for regulating gene-edited foods. As mentioned earlier, regulations on gene-edited foods vary widely between countries, with some countries treating these products the same as conventionally bred crops, while others subject them to the same regulations as GMOs. This lack of consistency can create confusion for consumers and producers alike, and can make it difficult for companies to navigate the regulatory landscape and bring new gene-edited products to market. Another challenge is the difficulty of distinguishing between gene-edited foods and GMOs. Some argue that gene-edited foods should be treated differently than GMOs, as they do not involve the introduction of foreign DNA into an organism. However, it can be difficult to distinguish between these two types of products, especially as gene editing techniques continue to evolve and become more complex. Finally, there is a need for robust safety assessments to ensure that gene-edited foods do not pose a threat to human health or the environment. While gene editing techniques like CRISPR-Cas9 are highly precise and targeted, there is still a risk of unintended consequences or unforeseen long-term effects. Regulators must work to ensure that gene-edited foods undergo rigorous safety assessments before they are approved for consumption, in order to protect public health and the environment. Regulatory challenges associated with gene-edited foods are complex and multifaceted, and will require careful consideration and collaboration between international organizations and regulatory bodies in order to establish a consistent and effective regulatory framework that promotes innovation while ensuring the safety of these products. 6. Conclusion. The controversy surrounding gene-edited foods highlights the complex ethical, social, and regulatory issues that arise when new technologies are developed that have the potential to transform the food system. On the one hand, gene editing offers the promise of new and innovative solutions to some of the biggest challenges facing agriculture today, such as climate change, food security, and pest resistance. On the other hand, there are concerns about the safety, environmental impact, and long-term effects of these technologies, as well as the potential for unintended consequences and the creation of new inequalities in the food system. Moving forward, it will be important for regulators, industry groups, and the public to engage in a robust and transparent dialogue about the benefits and risks of gene-edited foods, and to work together to establish clear and consistent guidelines for the development and regulation of these products. This will require collaboration and cooperation between countries and international organizations, as well as a commitment to ongoing research, safety assessments, and public engagement. The development and regulation of gene-edited foods must be guided by a commitment to promoting the public good, protecting human health and the environment, and ensuring that the benefits of these technologies are shared equitably and sustainably across society. Gene-edited foods represent a rapidly evolving field that has the potential to transform the food system in significant ways. By using precise and targeted gene editing techniques, researchers and industry leaders hope to develop crops that are more resilient to environmental stressors, more nutritious, and more sustainable. However, the development and regulation of these products also raise complex ethical, social, and regulatory issues that must be carefully considered and addressed. The controversy surrounding gene-edited foods highlights the need for a transparent and inclusive dialogue that takes into account the perspectives and concerns of all stakeholders, including consumers, industry groups, scientists, and regulatory bodies. To promote the safe and responsible development of these products, it will be important to establish clear and consistent guidelines for their regulation, as well as to prioritize ongoing research and safety assessments that take into account the potential long-term effects and unintended consequences of these technologies. The success of gene-edited foods will depend on our ability to balance the potential benefits of these technologies with their potential risks and limitations, and to ensure that the benefits of these innovations are shared equitably and sustainably across society. By working together to establish a responsible and effective regulatory framework for gene-edited foods, we can harness the power of these technologies to create a more resilient, sustainable, and equitable food system for all. Thanks for watching.